Starting early on in the semester is a good thing because it's going to let you glide to the latter part. The ones that just end up starting at the end, right before the test, they're freaking out, and then it's not as much fun. I know that's the general spiel that I give to students and everybody's going to give, but when I actually followed that in Cairo school, it was much more fun that way. And my grades were really good, and so that helps. So, but today we're going to do some work on the cell. When we study cells is fundamental as they represent the smallest living things on Earth. Knowing how they work helps us better understand the function of the human body. So these are the smallest living things that we have. What is the study of cells called? Cytology. That's pretty fundamental. That's just the word. And ology is the study of. So cytology, cyte means cell, and then the ology is the study of something. Pathology, physiology, and cytology, histology would be addition to do that. So you know these science people, they all have to come up with these names. They feel better about that. Some organisms, many bacteria for example, are unicellular, which means that they are made up of only one single cell. We, on the other hand, have about 75 trillion cells that make us up. What is that called, that kind of organism? Multicellular, right. So that's that. So 75 trillion cells, that's a lot of cells. That's 75,000 trillion cells. That's just, it's unconceivable for me how big that number is. So if it is for you, that's fine.
Not all our own, right?
No, these are our own.
Humans?
These are the bacteria stuff. Well, but if you think about it, a third of that is red blood cells, right? So that's a lot of cells that go right into that department that don't even live that long. And then 100 billion we have in the nervous system. That's also a pretty big number right there. But yeah, that's true. We have a lot of cells. These are 75 trillion are the cells with our own DNA, our own cells. We have a lot of cells, of microbial cells in us and around us, on top of us, which is part of that whole world. So we do have a lot of cells that are ours. And it's very important that we know that these microorganisms, a few years ago it was like, ooh, that's gross. And now everybody sort of understands that better in the real world because we use probiotics and stuff like that. But it's very, very important that we're knowing that most of the microorganisms have helped us to become who we are. They're not pathologies, they're not diseases. And so when we just go with the generic Lysol wipe all over the place in the house, we've got to be really, really careful if that even makes sense. Because it's not, it's not disinfected anyway. And we got to, and they have studied that children, for example, who grow up on a farm, have better immunity than inner city kids. And that has to do with the exposure of things. What do we use? And since it's all here anyway, we got to think about that. Particularly after kids. A little dirt goes a long way. Cell size is measured in microns. That is a length measurement indicating something is very, very small. A millimeter, which is a 25th of an inch, which is like in between my fingers, it's tiny. Take a ruler out. That making a millimeter is about 1/25th of an inch, making a micron about 1/25,000th of an inch. So it's just very, very small. We can see, with the naked eye, we can see about a human egg, which is 120 microns. The question is, the question often asked on MCATs and stuff like that, how large is a red blood cell more or less? About 5 to 7 microns. I don't ask questions on numbers. In this respect, but I brought that up because it is one of those that comes up in these standardized tests.
What was the range?
5 to 7, I believe, right? Or 5 to 8. 5 to 8. And then the machines here, these cells, they come in many different shapes. And when we talk about shapes, we always talk about that structure dictates functions. And what does that mean, really? Give an example, for example. Anybody? So yeah, go ahead. I know it's a little weird. This one is one that I might have to change the question, but.
Well, the ova, rounded edges make it good for traveling. You don't ruin. It fits and it doesn't damage the kind of the system it travels through. Right.
That's an example. For example, the red blood cells are like weird, they're like frisbee-looking things. They're always wider on the outside. And do we have a red blood cell somewhere? And then they go indented into the inside a little bit because the nucleus is sucked out of them. And that gives you the shape that, number one, they're flexible, they can squeeze through small channels like capillaries. And number two, the distance to any place on the inside is not as long, so the oxygen which goes in here, that is carried around by the red blood cells, has an equal distance to travel. And so it doesn't have to travel all the way from the outside to the nucleus that long, because it wouldn't be efficient. So that's just another place where we can see shape. Or a muscle cell is a long sort of cylindrical, weird-looking cell or spindle-looking like, and that can contract. And so that's shape determines function. How something looks is what it can do. So one good question you ask when you see something is that question, what could that do? A heart. What is a heart? That's an whole organ, but what does it look like? It's got 4 chambers. What do you think it's going to do? And then from that perspective, asking the questions answers a lot of the mechanisms and the functions deeper than when you just sort of like read it, look at that, it has 4 chambers, it pumps water, it does this. You don't ask those questions. So shape is a central piece when we talk anatomy and physiology. An organism is made of many different types of cells. Some are muscle cells, some are nerve cells, some are red blood cells, etc. An organism starts out with one such cells. In human, it's a fertilized egg that begins that multiplying process. Daughter cells adapt their size and shape to whatever task they are ought to accomplish. What is the process called that makes these cells unique? Somewhere back there. Hello. What is that called? Did you guys do these things, the questions? Well, I get the email. I just counted them all, and I put some marks behind the people who sent me the email. But what is that process called? Anybody? Differentiation. So we make the cell different to what its task is. That term is differentiation. We can also call it specialization. Sometimes they use that term as well for that. Each cell has the same basic structure. It has a boundary-given membrane surrounding a liquid inside. The liquid inside is known as the cytosol. Internally, a large dark sphere in the center, which is the nucleus, holds all the DNA, while other smaller solid structures with specific functions float around. Those other smaller structures are known as organelles. Remember that, like little organelle. It's a little organ. What is the cytoplasm?
It's the area of the cell, including the cytosol, but excluding the nucleus.
That's right. Cytoplasm is, well, let's go this. The watery stuff is the cytosol, mostly, with some protein in it. Then we got so we have cytosol, then we got a nucleus. And then we got organelles. And the cytoplasm is the cytosol plus the organelles without the nucleus. That's just a definition thing. Alright? Then we also have the cell membrane. And I think we're going to start talking about the cell membrane first. Because we talked about that thing in chemistry, when we talked about lipids a little bit. There's like a certain specific type of lipids that can make up a cell membrane. Let's see where we got that thing. The cell membrane surrounds the cell. It creates a boundary between the inside and the outside. Since we are mostly water, a great medium for that is an oil film around the entire cell. That way, only oily type substances can pass through it, but watery type substances cannot. Well, some of them leak a little bit, but generally speaking, they cannot. So, when we go back to chemistry, we had the hydrophobic elements can go and penetrate through. Hydro means water, phobic means fearing, so that's the oily stuff. And the hydrophilic elements, which are the watery substances, hydro means water, philic means loving, they won't pass through generally. And so, how do we make an oil film? What chemical molecule that we've learned makes up that membrane, makes up that oil film? Anybody remember? It's a phosphate. So it's a phospholipid. The phosphate is one part of it. So we have that phospholipid bilayer that makes up that cell membrane. So when you look at that here, this is a good sort of look. The top word here says hydrophilic, bottom word here says hydrophobic. So when you look at a cell membrane, you got these round structures. These are the phosphate groups, and they are hydrophilic, they're watery. And then you got these tails, these two tails going down, which are actually the same structure as in a triglyceride, those three chains coming out of carbon chains. Because just here we only have two, because one of the backbone places is taken up by the phosphate. So this, again, the molecule is great because the outside is water loving, the inside is water fearing. I take two of these molecules and point the hydrophobic end towards one another. Then I have a membrane that the top is water loving, the bottom is water loving, but the inside is not. But that's an oil film. So that is very, very helpful because in there we then have this boundary that keeps stuff out that we don't want.
What would be an example of something that you don't want?
Well, like you don't want just glucose to go in and out, for example, because that's water loving. The same as proteins. You don't just want proteins to leave the cell from the inside out if they need to be inside the cell. So it just gives you that boundary, and then what we do for the things that we want to get in and out, we have channel proteins. So these are structures like, where's the channel one? Right here's the channel one. See, that's a channel protein right here. So the molecules can go right through. If this is a glucose, for example, that can go right through when this channel protein is open. It is only open when an insulin is attached to it. Otherwise, it's closed. And when it's closed, that glucose that bounces back, it cannot go in. And so that's very helpful. If it were just letting it through, it would be all over the place. No, there wouldn't be any distinction.
So if the outside layer is water loving, but it's not dissolving, right?
No, they're not dissolving.
What's happening when you dissolve? Is that not
Well, if you look dissolve from our perspective, when we look at what bonds would dissolve in water, for us that was the ionic bonds.
right?
So we get the ions created. And we don't want these ions to go in and out. They are stuck, they're watery.